Here at ESMO 2022, I presented the second interim analysis of overall survival for the TROPiCS-02 trial. And this data showed that overall survival was significantly longer with sacituzumab govitecan versus treatment of physician choice, a menu of chemotherapy agents, in patients with heavily pretreated hormone receptor-positive, HER2-negative metastatic breast cancer. The median improvement was 3.2 months. We also showed a significant improvement in overall response, as well as clinical benefit rate and duration of response, and a delay in deterioration of global health status, quality of life, and fatigue with sacituzumab. And this contributes to our previous presented and published data showing a significant improvement in progression-free survival with sacituzumab govitecan in this patient population. So we've made unbelievable advances in the last couple of years with antibody drug conjugates, and this year in particular, we've been able to expand the use of antibody drug conjugates into hormone receptor-positive disease. Sacituzumab govitecan already showed an improvement in progression-free and overall survival in patients with heavily pretreated triple-negative breast cancer, a group of patients where we were severely limited in treatment options that even improved response, much less PFS and OS. So we're very familiar with sacituzumab govitecan and in managing the toxicity. So what's happened this year is that we've had a trastuzumab antibody drug conjugate, trastuzumab deruxtecan, with also linked to a topoisomerase inhibitor, very different antibody compared to the TROP2 based ADC that sacituzumab is, show an improvement in PFS and OS in patients who have HER2 low, largely hormone receptor positive, metastatic breast cancer. These patients are less heavily pretreated in DESTINY-Breast04, the trial that showed this, with a median of 1 line of prior chemotherapy for metastatic disease, and not everybody received a CDK4/6 inhibitor, compared to TROPiCS-02, where there was a median of 3 lines of prior chemotherapy. In addition, in DESTINY-Breast04, these patients had to have centrally confirmed HER2 low disease, defined as 1+ or 2+ without gene amplification. So now we have 2 drugs that have shown improvements in PFS and OS, but thankfully they're in different patient populations, so we could think a little bit about how we would use these agents in clinical practice. In triple-negative disease, I would still use sacituzumab first because there's a phase 3 trial with data and just a small cohort that was an exploratory cohort in DESTINY-Breast04 in patients who had HER2 low disease. In patients who have hormone receptor-positive disease, also HER2 low, T-DXd, or trastuzumab deruxtecan, is approved in those patients who've received 1 line of prior chemotherapy, so that's the obvious first choice. Sacituzumab govitecan is a great choice for patients, a third of the hormone receptor-positive population, who have HER2 zero disease at present, although trastuzumab deruxtecan is being studied in the HER2 zero and very low population in DESTINY-Breast06 now. We have populations where we might use one ADC versus another ADC, as I just explained. But what about the patients who've already had one or the other? And I think the next step is exploring these agents in sequence, because the activity of these agents has to do with the antibody target as well as the toxin. These 2 ADCs have different antibodies, different targets, and although both of the toxins target topoisomerase 1, they're different toxins and have different toxicity profiles. So we may see, as we've seen in microtubule inhibitors, sequential efficacy by using these agents. So it's great to have a new menu of agents that improve PFS and OS. That's really unheralded. We have so many drugs we use that we don't have that data on in sequence in patients with metastatic disease. And of course, our next step is to try and move these agents earlier in the course of therapy to try and help patients live longer and potentially have less metastatic disease.